Hello and welcome to part 3 of OpenAI Cadence Molecular Sciences webinar series on the essentials of computational chemistry. Today we are talking about molecular dynamics, simulations, and relative binding free energy predictions in the context of drug discovery. And I, Lukas Eberlein, application scientist at OpenAI in Germany, have the honor to present to you today on behalf of Paul. As with the previous webinars, you can let us know where you're joining from in the chat. For today, we have changed the Q&A format and will do it as a discussion between our MD experts, Chris Neal and Christopher Bailey. Please add any question you have to the Q&A section in the interface, and we'll answer as many questions as we can during the discussion. Today, out of necessity, we aim to provide you a wide but shallow overview of problems and solutions in molecular dynamics simulations for binding affinity. The field is broad, and there are lots of interesting things I don't have time to cover today, but there's a lot of additional material we will make available together with the slides after the webinar. So if you want to dig more into certain aspects, please look at these resources. Before we get into the technical aspects and the exploration of the science, I want to briefly explain what we do in computational chemistry. And that's primarily supporting drug discovery. We want to get drugs into the hands of patients who need them as efficiently and cost-effectively as possible. Here's an overview of the drug discovery process as a series of phases. It's a lengthy process that can take more than 10 years and costs over $2 billion for an average drug, and it's extremely risky. Tens of thousands of molecules go in to guarantee that the drug comes out at the other end. So, what we are doing in this series and at OpenAI Cadence Molecular Sciences is looking for computational tools that primarily support the earlier stages of the drug discovery process, the first 4 parts here. But of course, anything that has to do with science, especially the science of human biology, is much more complicated than the idealized process shown here. The part of the process we are looking at today is 3, lead optimization. Here, a lead compound has already been identified using the techniques we've discussed in the first 2 parts of this webinar series. In this phase of drug discovery, the teams are usually concerned with balancing several properties of a compound series against each other to find a candidate that is suitable for nomination to the clinic. This includes not only affinity for the target of interest, but also permeability, solubility, selectivity, toxicity, and much more. The process of lead optimization is inherently cyclical, and these sequential cycles consume time and valuable resources. The cycle usually consists of 4 phases: design of new compounds, making them, testing and biological assays, and analysis, and it is therefore referred to as the DMTA cycle. Computational chemistry can help to speed up the design part of the cycle and make it more efficient. Today, we focus on molecular dynamics methods for lead structure optimization, a sophisticated physics-based way of modeling. An important aspect of molecular dynamics is the introduction of time as a factor in molecular modeling. So far, the essential series has dealt with static molecular representations, 2D or 3D, but with the addition of time, we are able to study the dynamic evolution of systems over time. What is such an MD simulation? We simulate and analyze the physical movements of atoms and molecules. We gain access to the dynamics of a system by applying Newton's laws of motion to all atoms in the system, and usually use a molecular mechanics force field as potential energy function. We describe our system at the atomic level, but there is no reactivity in MD. We do not break or form covalent bonds. And perhaps most importantly, an MD simulation allows us to calculate thermodynamic properties of our system, such as protein-ligand binding affinity. Here, you can see an example result of an MD simulation. We see a video of the MD trajectory, in other words, the evolution of the system coordinates over time. But what exactly do we see here? We have a protein, a KRAS mutant, which is involved in a large proportion of human cancers. And there's also a small molecule, a drug developed by Mirati Therapeutics to treat patients with this mutation. We can see that a small pocket opens here on the right side of the protein and that the ligand binds there. Here I show 20 nanoseconds of the simulation derived from an overall 60 microsecond MD trajectory. Such a run requires 3 days of compute time with up to 100 GPUs and costs around $250 to $400 in cloud computing. 2 lessons here. First, we learned a lot about our system. We could see where and how our compound binds and even observe pocket formation. Second, we spent a lot of time and money on a single simulation. During this seminar, we will discuss how to efficiently use MD to study protein-ligand interactions in drug discovery projects. If we want to be able to study molecular systems with MD, we need to know the time scale of the motions underlying the observable of interest. Observable based on local flexibility can be observed by sampling nanoseconds, but more global or collective motions such as protein folding or ligand binding require orders of magnitude longer simulation time. This corresponds to a much higher computational cost, so capturing some of these processes could take decades with current hardware and is therefore not feasible. Of course, the simulation time also depends on the size of the protein and the MD engine used. We will discuss later how to gain access to these observables in a more clever way. Here are some problems MD can solve in drug development. MD helps us to predict and validate protein binding sites, which is very important for drug development. We need to know where and how a ligand binds so that we can tailor its properties to make it an effective drug. Proteins are not static. We often start with a crystal structure, but that's only a single point in the conformational landscape of the protein. MD can help us explore this conformational landscape and how the protein acts in the body. And of course, protein-ligand binding affinity. MD can predict how certain changes in our lead compound will affect the binding affinity and, in this way, speed up the DMTA cycle, as with good predictions, fewer molecules need to be made and tested. Today's focus is on MD to study protein-ligand interactions. So, to do MD, we need a protein and a ligand. We're going to spend some time today talking about the preparation of the protein to get it ready for MD. The ligand should be appropriately placed in the binding pocket of the protein, as discussed in the second part of this series. Protein-ligand complexes need to be placed in a simulation box in which we place the entire simulation system consisting of the complex and its environment. And we need a method to calculate the intra- and intramolecular interactions, which is usually a force field, but QM or hybrid approaches are also possible. I won't talk too much about ligand preparation for MD, as Paul has discussed this in detail in the first 2 parts of this series. Basically, there are 2 scenarios. Either we already have a ligand structure in pose, for example from a crystal structure, or we don't. If we need to generate a ligand structure first, we'll need to generate conformations as discussed in part 1 and accurately predict the poses as discussed in part 2 of this series. Since docking does not always lead to good poses, there are specific algorithms for pose prediction, and we can use MD to validate these poses and assess their stability. In both cases, we need to make sure that the protonation state of the ligand is correct. And we need to calculate the atomic charges with high accuracy. Methods we can use to generate atomic charges include RESP and AM1-BCC. Here I would like to briefly acknowledge my colleague Christopher Bailey, one of our 2 experts in the Q&A session, who is an expert in this field and has developed several widely used charge models. I want to go more into the details of protein preparation, because all protein structures are models and are built to explain collected data. The job of the structural biologist is to use software and experience to fit atoms to the data, the measured electron density, to create a model, so the atomic coordinates that best match the data. We should be able to assess the quality of such a model so that we can correctly evaluate the results from our MD. The quality of the model is a key determinant of the accuracy of the result it can provide. So it's crucial that each model building step is carefully designed and evaluated. The easiest way to check the quality of a structural model in the PDB is to use the PDB's own validation report. This is such a report for 2 chicken lysozyme structures. Resolution has often been used as a measure of model quality for X-ray structures, the method we are focusing on today, but several measures are given here. The middle group here essentially assesses coordinate quality, while the others provide information on the global and local fit to the experimental density. Looking only at resolution, one would opt for the top structure. However, looking at all the metrics, one could argue that the second structure has better coordinate quality and better matches the experimental density, both globally and locally. The resolution is lower, so less data was collected for the second structure, but the model fits the data much better. Here, both structures have the same resolution, but the model on the left fits the density much better than the poorly fitted model on the right. DPI is equivalent to error in measured solubility or log p-value. These data should never be reported without error, and neither should protein structure models. To simplify the assessment of model quality, the Iridium criteria were developed to divide X-ray structures into 3 classes before subsequent modeling. The 3 classes are highly trustworthy, moderately trustworthy, and not trustworthy. This classification is helpful in defining the user's expectations for modeling a particular structure. In general, not trustworthy structures should not be used for modeling unless there are no alternatives, and even then, the results should be taken with a grain of salt. Here we have a structure model from the PDB as we can download it. It's a CDK12 structure with a resolution of around 3 angstrom, which we can easily visualize in any viewer. This PDB file contains 4 chains, A, B, C, D. A, B, and C, D are 2 biological units. Each biological unit is a functional unit. Each biological unit contains 1 bound ligand. There are broken caps. We can see some obvious problems with this structure. There are no hydrogens and we have uncapped termini. But there are also some problems that are hard to spot by just looking at the structure. Some missing atoms and side chains, sometimes alternate locations, clashing atoms, and some, but not all, waters. Unfortunately, this structure is not suitable for modeling with MD. We will now go through the typical steps required to prepare such a structure for subsequent modeling with MD. First, we need to retrieve the data, ideally the model and the raw data. To become aware of possible problems, we can use information from the PDB header and compare the model to the experimental density. This helps to identify missing residues and more general geometric problems. We need to generate and compare the biological units and our unit cell. They can be identical or in different conformations, so we need to pay attention to whether we need to proceed with one or both units. We need to identify and split the components in our system. Usually a system contains more than just the macromolecule and the ligand. There are solvent molecules, cofactors, especially metals need to be handled carefully, ions, sugars, artifacts from the crystallization process, and much more. All these components can be important to the system, so we must consider them all and decide which ones to include in the simulation system and how to treat them. Sometimes metals need to be constrained to parts of the protein, and some of the components need specific force field descriptions. We cannot model a protein that is missing parts. This is a big difference to preparing protein structures for structure-based design. For MD, we need a complete protein chain and therefore need to model missing side chains. In addition, the protein chain must be kept. This is done by adding an acetyl or amine to the ends of the protein chain. Loops are often crucial for the function of proteins as they can play a role in regulating their activity. They can be very flexible, especially in the absence of a ligand, and are therefore often poorly resolved in experiment. Modeling their conformation is difficult but important. Missing loops can be modeled knowledge-based by using known conformations for the missing sequences, by using reference structures, and even ab initio methods are in use. Hydrogen is not resolved in X-ray experiments. To resolve it would require Newton refraction, but this is unusual. Therefore, we have to model hydrogens. The greatest attention must be paid to the binding site of the protein, as the placement of the hydrogen is crucial for the hydrogen bonds between protein, ligand, and water. The typical workflow is shown here on the left side. Normally, the hydrogen atoms are placed in such a way that the H bonding network is optimized. To do this, The ligand protomers are enumerated and the side chains are rotated so that the hydrogen bonding interactions are maximized. For docking or posing, a receptor grid could be added, but it is not needed for the MD itself. Finally, we should evaluate our model. The Iridium criteria are helpful here, as is a look at the density coverage of the ligand. You should also examine the hydrogen bonding network in the binding site. To conclude this section, I would like to highlight some of the differences in structure preparation for docking and MD. For MD, we need the full protein chain and need to model missing loops or broken chains, while for docking, the full binding site is needed, but missing residues and loops far from the binding site can be ignored. We need to add hydrogels and optimize the H-bonding network, cap the protein ends, and assign partial charges to the whole system. By using these robust modeling approaches, we increase the quality of the models and the likelihood of generating accurate predictions. Nowadays, tools that perform the preparation steps automatically are usually used in practice. Manual control of the model quality is still necessary. A simulation system is more than just a prepared protein-ligand complex, and its structure has significant influence on the results. The protein-ligand complex needs to be placed in a sufficiently large simulation box. The box here is cubic, but that's not the only possibility. A lot of different shapes are possible. Usually, the shape is chosen to minimize system size so that the MD runs more efficiently. We need to solvate the system. There are various tools that can be used to place solvent molecules in the box in such a way that the desired density is achieved. Using a finite box would result in surface effects dominating the system. In such a box, there would basically be no bulk water. Instead, water-protein and water-box surface interactions would dominate, so that no bulk properties could be derived. Increasing the size of the box is not a good solution to this problem, as it would lead to a higher simulation effort just to simulate additional water molecules and not to gain additional knowledge about protein-ligand interactions. To solve this problem, we can apply periodic boundary conditions. We create an infinite system by replicating our system in every direction in space, as shown here in 2D. This means that an atom that leaves the box to the left re-enters from the right and keeps all its dynamics. And we need to think about charges. With an infinite number of replicas of our system, it should be net neutral, which can be achieved by adding counter ions. We can add more salt to better mimic physiological conditions. Water, the most used solvent for biophysical simulations, has a significant impact on our simulation system. Some of the most important influences on protein-ligand interactions are solvation and desolvation of the components, the influence of thermodynamically stable or unstable waters, and water-mediated protein-ligand contacts. Water is a complicated liquid and not easy to model, so we should briefly discuss how to treat it in MD. An implicit description does not require sampling of solvent configurations and is therefore computationally efficient, but since the solvent is usually described as a dielectric continuum, it does not sample explicit solute-solvent interactions. There are many models for explicit solvent molecules. Standard models are TIP3P and SPCE, which are 3-point waters. They consist of both hydrogen and the oxygen atom, with corresponding force field parameters. But there are also complicated models that use 4 or even 5 sites to improve the electrostatic behavior and the geometry of the water with additional dummy sites. Polarizable water models are also possible, but computationally less efficient. The parameterization of water models is non-trivial. Here we see how a change in the force field parameters changes the coordination number of water from a simulation. Creating a water model that accurately reflects most of the important bulk properties of water is a complicated multi-parameter optimization. In practice, explicit water models have become the standard method for most molecular dynamic simulations. The main reason for this is the atomic level insight that these models provide. Effects such as partial desolvation, water rearrangements, and explicit solute-solvent interactions are difficult to model implicitly and play an important role in drug development. Choice of the potential energy function influences the simulation result just as much as the structure of the simulation system. And here I will concentrate on molecular mechanics force fields. The force field consists mainly of two components, a functional form and a set of parameters. It is the potential energy function for our system. The potential energy function can be divided into two groups, bonded and non-bonded interactions. The bonded terms describe the interactions between covalently connected atoms. Non-bonded interactions apply to all non-covalently connected atoms. The bonded interactions include terms for bond stretching, angle bending, and torsional rotations. Corresponding parameters can be derived from fitting to quantum chemical calculations or spectroscopic data. Depending on the force field, additional energy terms may occur. The non-covalent interactions are electrostatic and van der Waals interactions. Electrostatic interactions are evaluated using the Coulomb potential, while van der Waals interactions are often approximated using the Lennard-Jones potential. The molecular mechanic force fields allow us to calculate an enthalpy-based estimate of protein-ligand interactions. The Lennard-Jones potential is a pair potential that is often used to approximate van der Waals interactions. Note that the repulsive part is too steep, which requires proper equilibration of our simulation system. The potential energy approaches zero as the distance between atoms increases, but it is asymptotic and will never be exactly zero. This means that the range is in principle infinite, and we would have to calculate the potential between all atoms in our system. But at the same time, the contribution after a certain distance threshold is negligible. In practice, cut-off distances are used. The potential is only evaluated for atoms within this cut-off distance. Van der Waals interactions are short-range, so this concept is much easier to apply to them than to the longer-range electrostatic interactions. Here I'd like to acknowledge my colleague Tom Darden, who has been instrumental in developing techniques for the efficient calculation of long-range electrostatics and molecular dynamics, which has been crucial in making MD the useful tool it is today. We have to balance the size of our simulation box and cutoffs so that an atom experiences at most one image of every other atom in the system. Careful preparation of the simulation system and the choice of suitable force field parameters are crucial for the accuracy of the simulation results. Here we have the general MD algorithm. It starts with the initial coordinates of the system. We must assign initial velocities to all atoms so that we can have a certain initial energy in the system and can calculate the forces acting on each atom. We can use these forces to advance the system. While time is continuous, we cannot advance our system continuously because we cannot solve Newton's equations of motion for a system with this size analytically. We must integrate these equations numerically to access a new set of coordinates and therefore discretize time and advance our system in steps. Here is the Verlet algorithm, one of the most commonly used MD integrators. Finally, we have to write down our new coordinates, velocities, forces, and so on, and we can restart the cycle until we have a trajectory of the desired length. We move the atoms forwards in steps of femtoseconds, so we have to repeat the cycle millions of times to simulate nanoseconds of motions. To ensure stable simulations, a protocol of minimization and equilibration is usually followed. First, the entire system is energetically minimized to resolve potential clashes in the initial structure and ensure that we have a good starting geometry. Then initial velocities are assigned, usually from a Maxwell-Boltzmann distribution at the desired temperature. Now the system must be brought to equilibrium. During the equilibration, initial constraints are often released over several steps and the temperature is gradually increased. For stability reasons, an initial equilibration is often performed in the canonical or NVT ensemble, which means that we have a constant number of particles, the atoms we placed in our box, a constant volume, the box volume, and we scale the velocities so that the energy in the system matches our desired temperature. The production run is most often carried out in the isothermal isobaric or NPT ensemble, which corresponds to physiological conditions. Therefore, a second equilibration step is often used, in which we keep the pressure constant by scaling the volume of the box. MD simulations are computationally expensive. The potential has to be evaluated for all atoms at each time step, and we must do millions of them with typical 1 to 4 femtosecond time steps. Constraining the smallest motions in the systems allows us to use larger time steps and can be done by applying algorithms like shake or rattle. Here we see the results of 3 MD runs of the same license line system plotted as solid, dashed, and dashed dotted lines. We see 3 observables: the root mean square deviation from the initial structure, the radius of gyration, and the solvent accessible surface area. The results are different for each simulation, showing the stochastic nature of MD, and that simulations take time to converge, even for these relatively simple properties. And so, we have to run very long simulations to get access to reliable data, especially for slower motions. With brute force MD, it can take years and compute before we can observe a single ligand binding event. So brute force MD is not a solution for many problems. Calculations can be sped up by using enhanced sampling techniques. We can manipulate the free energy landscape to probe certain observables in a more efficient way. Methods such as replica exchange, weighted ensemble MD, or the use of bias potentials allow us to sample protein dynamics with less computational costs when, for example, observe the formation of alternate binding sites and proteins. And alchemical transformations help us to calculate binding free energies as a routine task these days. Here we have a decision tree that helps us determine which enhanced sampling method is appropriate for which problem. You can see that there is a variety of enhanced sampling methods, and you need to carefully consider whether the one you choose is appropriate for your problem. We will link to literature that discusses some of the pros and cons of the methods in the supplemental material. I will discuss the alchemical transformations in more detail later in this presentation, but first I'd like to show you what enhanced sampling is capable of and discuss how we can analyze protein ligand MD. This is the trajectory of a weighted ensemble MD run, a method that allows us to accelerate sampling across a progress coordinate. We have a small molecule, saccharide pictured here, and the progress coordinate is the movement of this compound through a lipid bilayer. For the sake of simplicity, only the water molecules that interact directly with saccharide are shown. We can see that the molecule passes through the membrane, partially desolvates, and then solvates again when it leaves the membrane. Such simulations can help us to study rare events, such as the molecule passing through the membrane, and enable the calculation of kinetic rate constants, such as the permeability coefficient, while also providing mechanistic insight at the atomic level. Just as important as correctly setting up and running an MD simulation is correctly analyzing the results. We will discuss such an analysis in the context of lead optimization. In the drug discovery process, we narrow this funnel more and more. We have screened millions or billions of molecules to find a good lead and need to explore space around that compound to optimize all important properties. So we are now working with maybe up to thousands of compounds, too many for extensive manual analysis, but few enough to apply highly accurate physics-based methods like MD to them at a reasonable computational cost. The focus of the analysis will be on pose stability. A compound in an unstable pose will either not be a good binder, or we have not captured the correct binding mode with our posing method. Here we see the trajectory of the binding event that I showed earlier in the presentation. We can clearly see an interaction, but what do we learn from this? How do we quantify these results in terms of binding affinity or pose stability? MD analysis is time-consuming, serial, and somewhat requires the user to know what they're looking for. We need to do more than just looking at trajectories, especially when running MD on hundreds or thousands of molecules. As a further step in the examination of a trajectory, graphical analysis, such as this one, can be performed, which give semi-qualitative indications of the mobility of the molecule by coloring the ligand and protein residues according to the movements shown by them over the course of the trajectory, torsional changes that drive this movement, and the persistence of molecules' interactions with the binding site residues. Such an analysis can be done automatically and is an excellent way to distinguish between stable and unstable binding poses without visually inspecting all the trajectories from the compound series. The analysis of post-stability can be taken to a more quantitative level by calculating the interaction energies, for example at the MMPDSA level of theory, or by quantifying the stability of protein-ligand interactions over the course of the trajectory. These quantitative metrics can be used to approximate binding affinities and provide useful guidance on which compounds to move forward in your lead optimization project. Here's an example of a retrospective analysis of a compound series using OpenEye's binding interaction score. In this analysis, we know the experimental affinities and we can see a reasonably good correlation between them and the prediction with a rank correlation coefficient of 0.5. But we can also use such simple scores for the prospective analysis of MD results. At the top right, we again see the plot of the calculated binding interaction scores with both compounds 58 and 47 highlighted. The compound on the left has the best and the one on the right the worst binding interaction score. From the two-dimensional plot, we see a lot of movement in the MD for the right compound, much less for the other. More stable interactions, as measured by the score, and less movement in the binding pocket indicate that the left compound is more stable in the binding pocket. We can confirm this by looking at the actual trajectories. This shows that the automated projection of MD observables and the calculation of simple scores complement each other and can help to quickly assess the stabilities of our poses even in a larger series of compounds. Another important aspect when analyzing MD trajectories for drug design is the analysis of water. We have a variety of methods to choose from. Examples are GIST, WaterMap, SHMAP, or 3D RISM. To illustrate how water information can guide the design process, we can take a look at this example system. We have a structure from the PDB with a co-crystallized ligand. We see the results of the water analysis and that there is an unstable water in the immediate proximity of the ligand. On the other side, we have the same water analysis from the APO structure mapped onto the ligand, which is colored by the water thermodynamics in the respective parts of the pocket. The unstable water is clearly present, regardless of whether the analysis is performed for the APO or whole structure. When we look at the same protein with a different ligand, this unstable water molecule has been replaced by adding a methyl group to the ligand. This increased the potency of the ligand by more than an order of magnitude. This clearly shows how we can use water information to guide our design process and get new ideas. Back to the lead optimization cycle. MD simulations can help us to triage molecules by post-stability and provide useful information on water thermodynamics that can lead us to new design ideas. These methods are well established and can be applied to up to thousands of molecules. The remaining molecules can be transferred to more computationally intensive methods, such as relative binding free energy calculations. The purpose of these calculations is to prioritize the molecules to be synthesized by the medicinal chemists for testing in biological assays. Therefore, these methods must be able to rank compounds by affinity. Brute force estimation of binding affinities can be done by running very long simulations and observe the binding and unbinding. This way, on and off rates can be calculated and converted into binding affinities. These simulations could be an all-in-one solution, as you could also learn a lot about other interesting properties such as the binding pose and even potential alternative binding sites if we sample long enough. But for strong binders, like we have in lead opt, off rates are too slow to sample, and this approach is generally not useful. Like in other enhanced sampling methods, we will use a clever trick to calculate binding affinities in a more efficient way. This is a thermodynamic cycle for protein ligand binding. Simulating the binding process directly lets us calculate the reaction on top. Our trick is to simplify this cycle while avoiding the expensive simulation of the binding event itself. We achieve this by making an alchemical change to the molecule in the active site of the protein. Here we transform one ligand in the active site into the other by attaching a group to it. This allows us to calculate the relative binding free energy, which is the difference in binding free energies between these two molecules, and helps us to prioritize one of the molecules within this. In principle, it's also possible to make the entire molecule appear or disappear and gain access to the absolute binding free energy, but this leads to a larger perturbation of the system, which requires more sampling and is also less accurate because we can no longer benefit from error cancellation. How does the alchemical transformation of the ligand from left to right change the thermodynamic cycle? It is simplified. We only need to calculate the vertical path of the cycle to access the relative binding free energy. If we already know the binding free energy of compound A or B, which is often the case in lead optimization study because most of the time we already have some crystal structures, biochemical assets for the lead compound, we also have access to the absolute binding free energy for the other compound. To perform these alchemical calculations, one compound must be converted into the other. Usually, this is done by gradually transforming the molecule using a coupling parameter lambda. In this example, lambda for the phenol on the left, 0, and the compound is transformed in several steps so that lambda equals 1 for the benzene on the right. During this process, the hydroxyl group slowly disappears while the hydrogen appears. There are 3 established approaches commonly used in industry for the calculation of relative binding free energies: thermodynamic integration, free energy perturbation, and non-equilibrium switching. TI and FEP are computationally expensive because they require an equilibrium sampling of all lambda states, including unphysical intermediate states. NES overcomes this issue because many transformations are carried out and the ligands are quickly switched from state A to B out of equilibrium. The relative binding free energy is then calculated from the work distribution obtained from all these rapid switches. Since non-physical intermediate states play an important role in chemical transformations, we need to treat them correctly. Often, the number of atoms and connectivity in molecules A and B are not identical, so we cannot simply change the force field parameters from one atom type to another. We have to create a so-called chimeric molecule. There are 2 common approaches for doing this: a single topology approach shown on the left and a dual topology approach on the right. The single topology converts from one atom type to another. Dummy atoms are used when there is no corresponding maximum common substructure match between the 2 molecules for certain atoms. The dual topology does not convert one species to another, but only converts between dummy atoms and an interacting species, usually using soft copotentials to improve overlap between dummy and real atoms. To efficiently calculate RBFEs using these transformations, so-called edge maps have been developed to describe the perturbation network. An edge is an alchemical transformation between 2 molecules, normally done forwards and backwards as indicated by the arrows. Since all molecules are connected via the edge map, we can access the relative binding free energies of all molecules in our compound series. On the left, we have a star map, the minimal perturbation network. The central node is usually a crystal structure of known affinity. Another example is shown on the right. NOMAP usually has about 1.5 edges per ligand and shows several smaller closed cycles. This makes the RBFE calculation more robust because the additional cycle closure allows the correction of individual bad edges. It is also important for the stability and accuracy of RBFE calculations to maximize the overlap between species. On both sides, we have the same ligands in different poses. The ligands are poorly aligned on the left side of the image, which can lead to clashes or different binding modes after transformation and low accuracy. By contrast, they are aligned at their maximum common substructure on the right, minimizing system perturbation during the transformation. Not only bad alignments are problematic for RBFE calculations, also the role of water is very important to consider. Here we have a structured water analysis from an MB run and 2 ligands of congeneric series. In the static representation on the bottom, we see that we have a common highly structured water in both systems. On the left side, we also see a different, less structured water. If we transform the ligand from left to right, we can trap the less structured water molecule in its position and stop its slow exchange with the reservoir which can lead to collisions and low accuracy since we do not have room for two water molecules here as the fragment grows. In the opposite case, we shrink the fragment, creating space that cannot be immediately filled by water. This can also result in low accuracy from that edge. There are ways to account for this problem of trapped water. One of the most commonly used techniques is grand canonical Monte Carlo. This is how a typical result looks like. We have the experimental and calculated relative binding free energies for all edges. If we have a fully connected set of relative binding free energies for the edges, we can convert them to per ligand binding free energies. These binding free energies are typically reported together with some correlation metrics like linear and rank ordering correlation coefficient and some error metrics. Ideally, the slope of such a plot should be around 1, but that is not necessary for ranking compounds as long as the method correctly predicts a high affinity for the most potent compounds. Of course, we need to evaluate in each of our drug discovery projects if and how free energy calculations can help us make predictions. For this example system, we have to critically assess if we can base our decisions on the binding free energy prediction. While we have a good correlation and lower errors for this target, retrospective analysis shows that the free energy calculation correctly identifies the R groups of this molecule that leads to the most potent inhibitors. MD is great, but as for all methods, we should treat our results carefully and think about possible pitfalls. MD is a physics-based method with potentially high accuracy, but we always work with models, and these models might not accurately represent what we are trying to predict. We use force fields as our potential energy function. They are parameterized to capture the underlying physics, but are approximations and not always work well for everything. For example, torsion parameters are often refitted using quantum chemical calculations to increase the accuracy in RBFE calculations. MD simulations are stochastic by nature, so we should think about the time we need to run an MD simulation to achieve convergence and how many repeats we need to get statistically significant results. Also, the analysis of MD trajectories is non-trivial and oftentimes requires a lot of manual work. To sum up, I'd like to repeat some of the key points we've discussed today. MD introduces time and dynamics into molecular modeling. It's a standard method used in daily drug discovery work nowadays. It allows us to sample fast motions and protein ligand systems efficiently, but we need to introduce some approximations and use enhanced sampling methods to get access to biologically relevant slower motions. We always work with structural models in MD. We need to evaluate our models carefully before using them in a highly accurate but computationally expensive method like MD. But if we do so, we can learn a lot about our system. We can evaluate post-stability and lead optimization projects and gain access to design ideas guided by water thermodynamics. And the method is even capable to predict binding affinities of compound series with high accuracy. This is usually done using alchemical transformation and allows triaging of compounds based on binding free energies and can help us to accelerate the DMTA cycle. Before we move on to the Q&A discussion, I want to point your attention towards the next and final part of the series. Paul will talk about method comparison and decision-making on December 10th. If you're interested in using OpenEye Cadence Molecular Sciences tools in your drug discovery projects, please scan the QR code on top and get in touch with us. We offer free trials of our software. And if you are working in academia, you can apply for a free academic license by scanning the other QR code. And please subscribe to our mailing lists to stay updated on what's going on in the field. Thank you very much for attending today's session, and please stay a little longer for our Q&A session. Thanks for staying with us for our Q&A session. At first, I'd like to introduce our two experts. We have Christopher Bailey, who has worked in computational chemistry industry for over 30 years. After an academic training centered around force fields, he contributed the widely used RESP partial charge model to the AMBER force field. After that, he spent 18 years in drug discovery at Merck Frost, founding and building up the computational chemistry group and continuing partial charge model development, contributing the well-known AM1 BCC charge model. After joining us at OpenEye in 2011, He has continued developing various methods within OpenEye, as well as co-founding the OpenFF collaboration during his sabbatical in 2016. Since then, he founded and built the Affinity group at OpenEye, applying MD-based free energy methods to protein ligand binding, handing over his group to Chris Neal this past summer. And the other expert is Chris Neal, who leads now the molecular binding affinity prediction group at OpenEye Cadence Molecular Sciences. His interests include method development for rational drug design, cell signaling via membrane proteins, and uncertainty quantification. Chris is also an editorial board member at the Biophysical Journal. So we have several questions. I think this is a good one to start with. You talked a lot about ways that MD simulations can go wrong. How can I tell if they are right or wrong for my system? Maybe you want to start answering the question, Christopher?
Well, certainly that question has driven the science in protein ligand binding free energy methods for the last 30 years. Even when we're started, it's because although the statistical thermodynamics is rigorous, the way we want to apply it is always by gaining efficiency by trimming down the degree of effort we apply, most especially in terms of the time scale of ligand binding, which in that time scale slide that you showed earlier, Lucas, goes over a very long time, and we want to do our calculations in a very short time. So we apply many, many approximations to try and make our calculations more efficient. And this means that the beautiful statistical thermodynamics becomes compromised so that sometimes the calculations do not work the way we want them to. That is why they can go wrong. And we've always been trying to figure out ways that we can figure out when they go wrong. And so there are various metrics that have crept in over the years, like the overlap coefficient in lots of the free energy calculations. These various metrics are things that can tell us when something is wrong. But the issue is that even when these metrics can't find out that something is wrong, something might still be going wrong. So even today, as we apply these methods, which have gotten better and better, and these methods, these metrics can tell us when something is going wrong, we're still in the situation where we have to try out the free energy methods for the protein or the target of interest and basically get some experience on an individual target basis, and sometimes even on the chemical species that we're running on, the conformer series, to find out that our methods can work, for that project. And even now, there are some protein targets for which our methods will work and other ones where they just won't work, and we're still trying to figure out why. I think we're going to be still trying to improve ourselves on that front for many years to come.
Yeah, if I can just jump in, Lucas.
Go ahead.
Simple things people can do at the beginning, and one is just looking at the structural reorganization of the protein by, you could do a root mean squared deviation or something like this along the trajectory. If the protein is changing a lot, you could imagine running multiple repeat simulations and see that it's relaxing to the same basin. If it wasn't, then that would be cause for concern. And then you may have additional experimental data of whatever form that you can compare qualitatively or quantitatively to some of what you're seeing from simulations to gain some certainty.
Thank you so much. I see we have several questions about proteins. Maybe this is a good one to start with. Can I use an AlphaFold structure to predict binding free energies?
Right. If I can take that, Lucas, I'd say that's actually a large handful of questions kind of combined into one. You're asking if the structure is accurate, if it's stable in simulations, the structure of your target, that is. Can you identify the binding pocket from that model? Can you get good docked poses, sensible and accurate poses from docking or induced fit docking type methods? And then finally, the actual perhaps thrust of the question is, if those are all true, can you then get accurate binding for energy predictions? And there is some data in the literature now. Docking can be accurate from AI-based models, specifically AlphaFold, as can free energy calculations. they can also be inaccurate while not showing any clear sign of inaccuracy until you compare to experimental data. So there is the possibility of it working. You are increasing the probability of failure when you move to a homology model or an ML-built model. What it does do, though, is it provides an avenue for structure-based drug discovery where you would otherwise be limited to ligand-based drug discovery. And so first, it offers that opportunity, and it also offers a comparison. If you already have an ongoing ligand-based drug discovery effort for that target, you can, again, try and compare and see if you get qualitatively or quantitatively consistent results from, say, your 3D-QSAR ligand-based model and your structure-based model coming out from that ML structure.
And if I could just add to that, I'd say very much in the spirit of, you know, what you showed in the webinar, Lucas, and what Chris Niels talked about. I mean, the free energy calculations using MD, it's a high bar. It's putting a lot of demand on the model going in, as you explained in the section on building a good model. And so the AlphaFold, the protein folding model is primarily directed towards getting the correct fold and isn't necessarily going to put the same attention into getting all the side chains correct. To get a good docking, you need a good local structure around the immediate active site. But for MD, it's just a more demanding calculation, and the thermal motion across the whole protein ends up needing to have a good model at the level of the side chain placement as well. In the past, this has been a challenge for some of these machine learning approaches that can get the AlphaFold of the protein correct. And I think that as those methods improve how to place side chains, that will actually improve the ability of these models to actually, you know, be good inputs for these ligand binding free energy calculations.
Yep, we have a really related question. So, what can I do if there's not a crystal structure for my protein? I mean, AlphaFold could be one solution for this, but do you have any kind of opinions? What else people could try?
Well, let me just, especially on the heels of the previous question, I think one of the real disruptive developments over the past few years has been the ability of cryo-EM structures to actually be very competitive with crystal structures in terms of providing atomic resolution models suitable for protein ligand binding. That said, cryo-EM is a very different experimental approach to crystal structures, and it has its own challenges. And the cryo-EM structures can, fairly easily get sort of larger shapes and low resolution images, but when that information is good enough that you can actually get that degree of atomic resolution to get, as we said in the previous question, getting the side chains placed correctly, and the cryo-EM experimental structure can tell you when it has that degree of resolution, that at least for one can be a really good alternative to X-ray crystal structures as a starting point for free energy calculations.
And I'd like to add as well, we can also think back to the first part of this webinar series where Paul talked a lot about ligand-based design. So in case we don't have a structure of our system at all, but no an active compound, ligand-based design could, of course, be an alternative, but of course, very different method. Maybe in the interest of time, just one last question. Can I run free energy calculations for all my ligands from my virtual screening run? If not, why not?
Well, let me just start off with that, answering that question. That is, in principle, you could run it for all your ligands. If all our methods, in the light of our previous discussion on all the issues we have, if our methods all worked as well as they should with the physics and the statistical thermodynamics, we could run any ligands. But because of the many different directions that we've gone with free energy calculations, as you mentioned before, Lucas, you can run absolute binding free energy calculations, but they are, the trickiest of calculations, and that you could run on any ligand, but it would be very expensive and it would be slow, and it's very, very tricky. The most reliable calculations are the relative binding free energy calculations, which you talked more about. This is what most of the vendors are providing. This is what we are providing. And these ones require some degree of similarity as you move from one ligand to another ligand. So this actually places a constraint, kind of a loose constraint or a soft constraint or an approximate constraint on what kind of ligands you can run. For relative binding free energy calculation, the ligands need to be somewhat related with the methods that we are applying these days. How related they have to be, how close they have to be, that can be, again, target dependent, and you'd have to get a feel for that. But the normal, lead optimization, you have a lead series, it's congeneric, you're replacing R groups. This is something which we can, those are the ones which fall well into the category of the relative binding free energy calculations we can do routinely today.
Right, and so that similarity, Lucas, also, it applies to the ligand, but it also extends to changes in, say, the protein binding pocket target with different ligands would make the results, makes the computation more complicated and makes the chance of accuracy go down. Same for changes in, say, water structure around the ligand in the binding pocket.
Yeah, thank you so much to the two of you for being our experts for the Q&A session today. Glad that you have been here, and I did not need to answer all the questions myself. Thanks everyone for attending today's webinar and even staying with us for this Q&A session and asking all these interesting questions. And we hope to see you again for part 4 of the webinar series. Thank you.